Assalamualaikum. So in this lecture, what we will do is we'll talk about the third globular protein as an example of globular proteins. So first, in the first lecture, we talked about fibrous proteins, 3 examples. Then we talked about hemoglobin and myoglobin, and we got into more details about hemoglobin, regulation of its function, and pathologies related to hemoglobin molecule. So in this lecture, we'll go over immunoglobulins, and you can refer to as a main resource this lecture, as well as your biochemistry textbook. And there is a nice website on the NCBI that you can refer to as well. So we can divide types of immunity into 2 types. We have the innate, non-specific immunity, and we have the adaptive immunity as well. So the innate immunity is basically it's not specific. So it includes skin, and it includes phagocytic cells that eat everything that is foreign. And then we have adaptive defenses. These are more specific branches of immunity. So they do attack specific antigens like bacterial proteins or bacteria, viruses, very specifically. So let's start talking about B cells. So B cells represent a branch of adaptive immunity. Now, these B cells, once activated, they secrete molecules known as immunoglobulins. These are also known as antibodies. Now, what antibodies do, or immunoglobulins, is several things, but these are the 3 basic primary roles of immunoglobulins. The first thing is that antibodies can bind to pathogens, and they induce phagocytosis into immune cells. So it's almost like they guide these phagocytic cells to where antigens exist, so that these phagocytic cells, like macrophages, for example, they eat these antigens, and then antigens are degraded inside cells. Another mechanism is that they activate a system known as a complement system. What this system does, it's basically activation of immune cells that can release certain proteins, and these proteins can bind to the cell membrane of bacterial cells, and these proteins result in lysing or disrupting the plasma membrane of these cells, so they die. Now, something else is that antibodies do is that they can bind to antigens, bacteria, viruses, maybe molecules like toxins, and they neutralize them. So we're talking about 3 mechanisms then. We have phagocytosis, antibody recognition, activating the complement system, and neutralization. So these B cells, before being activated or before they bind to antigens, they have these antibodies, immunoglobulins, on the cell surface. And antigens, or a certain antigen, would bind to the antibody on the cell surface, and the cell that has the antigen bound to it gets activated. So the same cell starts to proliferate, they grow and divide, and they differentiate. They differentiate into effector B cells. These effector B cells produce the same exact antibody, so they're all clones of the same parental cell. They produce a lot of antibodies, and these antibodies are released. So it's almost 2,000 molecules are released per second, which is quite amazing by each cell. Now, so let's talk about immunoglobulins now. So immunoglobulins look like the letter Y. Okay, they are heterotetramers. So they're composed of 4 chains, and these 4 chains are different. So we have 2 identical chains and we have 2 other identical chains, but these chains are different than the little ones in there. Now these 4 chains or polypeptides are connected to each other via disulfide bonds. So you have the 2 big ones linked by disulfide bonds, and each little one is also connected to a big one also by disulfide bond. Now the other thing is that inside, within the same polypeptide, within the same chain, there are also disulfide bonds as well, stabilizing the structure of the molecule. Now, the antibody immunoglobulin can also be glycosylated, so they are considered glycoproteins, and mainly they are connected to the big ones. Now, these big ones are also known as the heavy chains, and the little ones are known as the light chains. So there are 2 heavy chains and 2 light chains. These 2 light chains are identical to each other, and these 2 heavy chains are also identical to each other. But the heavy chains and the light chains are different from each other. So by looking at immunoglobulins, scientists have noticed that these immunoglobulins consist of 2 domains. The first domain is known as the constant domain, and the other domain is known as the variable domain. So you can have 2 identical antibodies, you can have 2 antibodies that have the same constant domain, so it's constant, but they differ in the variable domain or the variable region. Now, each heavy chain consists of 1 variable region and 3 constant regions. So you have one here, second one in there, and the third one right here. On the other hand, the light chain, each light chain has 2 domains, 1 constant domain and 1 variable domain. Now notice that the variable domain of the light chain it interacts with the variable domain of the heavy chain, and the constant domain of the light chain is it interacts with the constant domain of one constant domain of the heavy chain. So as I said, you have antibodies having the same constant regions, but they differ in the variable region. Now something else is that you have what is known as the Fc domain. This is known as the Fc domain, and this Fc domain is important for linking, binding, associating the immunoglobulin to the surface of phagocytic cells. Okay. Now, right in the middle, there is a region known as the hinge region. Now, this hinge region provides flexibility to the molecule so that when it binds to the antigen, it can orient itself in a proper angle that it binds the antigen with high affinity. Okay. Okay. So let's look into the variable region now, because this is the most important part of the antibody. Now, the variable region is found at the tip of the Y shape of the immunoglobulin. It's the colored regions here in this rotating immunoglobulin. Now, this region, the part that binds to the antigen, is also known as the epitope. Okay, so the variable region contains the epitope, which is the antigen binding site. Now, each immunoglobulin can bind to two antigens of the same type. So this part right here, both the variable region of the heavy and light chain can bind to an antigen to a particular position or place on the antigen, and the other side of the immunoglobulin, the variable regions of the heavy chain and light chain, can bind to the same exact type of or position of another antigen, okay, but having the same structure. So what does determine? So this is the important part. This is the part that recognizes the antigen. So this part, the variable region, is different among different antibodies, and they differ by having different structure and different amino acid sequence. So there are 7 to 12 amino acids in each one of these variable regions that determine to which antigen an immunoglobulin would bind to. Okay? Now, what happens is that each B cell, by the way, can produce one kind of an antibody having the same exact type of a variable region. Okay? So once you have a B cell that binds to an antigen, it keeps producing the same exact immunoglobulin having the same exact variable region. Now, if we zoom into the variable region that is present right here at the tip of the immunoglobulin, there are specific regions that are also important, and these are known as the hypervariable region, also known as the complementarity determining regions, or CDRs. So from the name hypervariable, it means that the differences in the amino acid sequence exist actually right here in the hypervariable region. And the differences among antibodies at the hypervariable region is what provides both diversity in terms of binding to antigens and specificity as well as affinity. Okay, so once these hypervariable regions bind to the antigen, binding is usually strong, so there's high affinity. If we look at this hypervariable region even further, okay, so all we're doing here is that we're zooming into the variable region, particularly the hypervariable region, and now we're looking a bit further, zooming into this structure. And the hypervariable region actually exists as a loop. So there is this structure right here, and this loop is part of a larger structure that is full of beta strands. Okay, so you have beta strands in here. So this is a beta sheet structure, and extending to the outside you have these loops, and it's these loops that actually recognize and interact with antigens. Because of the way it looks, that is, you have these beta strands, and they go around in such a structure. They look like a barrel. Okay, so these beta strands are they exist in anti-parallel fashion. Okay, and it's almost like there are two halves. So you have like 4 in here and 4 in the back. But overall, they look like a barrel. So this is known as the beta barrel. Okay, but what's important is that it's the loops that interact with the antigen. So here's another look for the different heavy different structures or different domains of the heavy and light chains. So you can see here you have a domain, constant domain 1, constant domain 2, constant domain 3, so that, and here you have the variable region. That's okay, so that's the variable region right here. That's the immunoglobulin fold. Okay, this whole thing is the immunoglobulin fold, and extending to the outside you have the hypervariable region right here, the loops. Okay, same thing with the light chain in brown. There's one constant region and there's one variable region, and the same thing, you have the beta barrel structure and the loops that are extending to the outside. Now, so the antigen antibody binding is actually mediated by non-covalent interactions. So right here you have the antigen and you have the antibody, and the antibody can interact with the antigen via hydrogen bonds, electrostatic interactions, hydrophobic interactions, van der Waals interactions, and so on. Now, the affinity is determined, of course, by the amino acid sequence, as well as the length, the amino acid sequence within the loop, as well as the length of the loops. So overall, it's actually the three-dimensional structure that determines the antibody-antigen interaction. Sometimes, by the way, it's the amino acid sequence, and other times it's the three-dimensional structure of the protein. So it depends on the antibody and what it recognizes. Now, if you remember in the molecular biology course, briefly, I talked about diversity of antibodies, saying that we can produce a lot of different antibodies having different sequences. 10 to the 11th different antibody molecules can be produced, even more than that. And this is done by DNA rearrangement of the, if you remember, the exons of the joining region, the variable region, and there are different exons for the variable region. Imprecise joining of regions during the DNA rearrangement. You have addition, deletion of nucleotides during this rearrangement. Also, as B cells proliferate and as they synthesize DNA, they introduce mutations in certain regions, especially, of course, the hypervariable region. And these are known as somatic hypermutations. And of course, we have alternative splicing of the RNA molecule. So I'm not going to go over these again, these different mechanisms. What I care about in this lecture is for you to understand the basic structure of the immunoglobulin and how immunoglobulins can interact with antigens. Now, one level of diversity that can be created in immunoglobulins is in both the light chain and the heavy chain, and here I'm talking about the constant region, not the variable region. That is, for the light chains, there are two types or two genes for the light chains, and these are known as lambda and kappa. So that provides sort of like diversity, even though we're not talking about the part that recognizes the antigen. Now, as for the heavy chain, there are 5 types, and these are known as the IgA, IgD, IgG, IgE, and IgM. Ig, by the way, stands for immunoglobulin. And these are produced, each one of them, each constant region is produced from a certain exon in the heavy chain gene. And these exons are known as the alpha for IgA, delta, IgD, gamma, IgG, epsilon for IgE, and mu for IgM. So what happens usually when we're talking about the heavy chain, is that there's something happens known and it's known as class switching. So what happens first is that these B cells, by the way, contain on the surface IgM molecules and having a variable region. Now notice the order of the exons for the constant regions. You have IgM, IgD, IgG, IgE, and IgA. So what happens is that when a B cell having an IgM, when it recognizes an antigen, it gets activated. And then something known as class switching takes place. Now in this case, what happens is that the same variable region, instead of being linked to IgM, you have rearrangement of genes of the gene so that the variable region now is comes close to the exons of the IgG. So you have class switching from IgM antibody to IgG. And it can happen again, that class switching can take place so that the same variable region or the exons for the variable regions would rearrange themselves to get close to the alpha exon that produces the IgA. Okay, so this is known as class switching, and the constant region plays an important role in the type of immune cells that the antibody can bind to. So these are the different types of antibodies. We have IgM. What happens is that the IgM is produced from the mu constant region. Okay, it's basically expressed on the surface of B cells right before recognizing the antigen. So once the IgM on the cell surface interact with the antigen, the B cell becomes activated, and this B cell starts to proliferate and differentiate further, and it undergoes class switching. So what IgM does is that it promotes phagocytosis, that is immune cells eating antigens, and they also activate the complement system. And usually, by the way, the IgM appears as a pentamer. So you have so this pentamer, by the way, can bind to 10 different antigen molecules because each one of them contains 2, right? So you have 10 sites where antigens can bind to. Now, class switching takes place, and B cells start to produce the IgG from the gamma exon, and IgGs are monomers. They are the most abundant immunoglobulins in serum, and they promote phagocytosis as well as the complement system, and it's the only kind of antibodies that can cross the placenta. Okay. Now you have something known as the IgD. IgD is always on the cell surface. Maybe I should say, by the way, that IgM is always on the cell surface. It's not secreted. Now IgD is also on the cell surface, and it's not secreted. And the main function of IgD is really under investigation still. Now IgE is produced from the epsilon exon. It's also a monomer. It binds to the cell surface of mast cells. These are immune cells, and these are the ones that are responsible for allergies and allergic reactions. And then we have IgA. IgA is produced from the alpha exon. It exists mainly as a monomer or a dimer when secreted, and it is present in mucosal secretion. And it's the initial defense in mucus against pathogenic agents. Okay, now, so there are 3 terms that I would like you to learn. Antibodies can be idiotypes to each other. They can be isotypes, or they can be allotypes. So let's look at this slide, and you can refer to the previous slide, to read the information that I'll just explain in here. So antibodies can be isotypes. So meaning that you can have two antibodies with different concentrations, but the variable region is basically the same. Okay, this happens in case of class switching, right? So you have the B cell having an IgM on the cell surface. This B cell recognizes an antigen, and then the B cell produces IgM at first, and then it goes through class switching so that the variable region is now rearranged. So instead of being close to the mu exon, it becomes close to the gamma exon. So producing an IgG antibody, but with the same variable region. Now you can have allotypes as well. Now with allotypes, allo comes from the word allele. If you remember, allele in molecular biology is basically different forms of the same gene. So we talked about an eye color gene, and you have a green eye color gene, and a blue and brown and black and so on. So they are different forms of the same exact gene, right? Now with allotypes, we're talking about an antibody or immunoglobulins with the same concentration. But if you look closely to the concentration, there are individual differences between these immunoglobulins. So there are differences in the primary structure of the immunoglobulin that may result from single nucleotide polymorphisms, differences in DNA sequence, little differences in DNA sequences among individuals. So even though they have the same exact constant region, the same type, but there are little differences that can differentiate one person from another. And then we have idiotypes. Idiotypes is basically when we're talking about an antibody with the same constant region, but the variable region is totally different. Okay, so they can be produced against different antigens, or they can be produced against the same antigen, but the amino acid sequence of the first immunoglobulin is different than the other at the hypervariable region. So you can have one interacting with the antigen with higher affinity than the other one. Now, I'll finish by talking about monoclonal antibodies and how they are produced. Now, monoclonal antibodies are really important nowadays in treating patients of different diseases and diagnosing diseases as well. So what we are talking about here is that we're producing the same exact antibody, okay, from a B cell. So the B cell becomes immortal, meaning that it does not die. It keeps on producing the same exact immunoglobulin, and it survives for a long time. Okay, so it's almost like a cancer cell. And these B cells, these type of B cells, are now used a lot in pharmaceutical industry. So how do we produce monoclonal antibodies? By creating what is known as a hybridoma cell. So a hybridoma cell is basically a cell that is a hybrid, hagin, a hybrid of two cells. One is the B cell, and remember, each B cell produces the same exact immunoglobulin, right? So you have merging or fusing of a B cell with a cancer cell, producing a hybridoma cell. Now, this hybridoma cell would be immortal because it behaves like a cancer cell that keeps on dividing, and it behaves like a B cell producing immunoglobulins. And again, it's the same immunoglobulin. So this is why these antibodies are known as monoclonal antibodies, because they are exactly the same. So if you have a person, let's say having an antigen, now different B cells can recognize this antigen, and each one of them would be activated, and each one of them would produce antibody, right? The same immunoglobulin for each one of them. But these different antibodies are different. They have different structures. They have different sequences. These are known as polyclonal antibodies because they are produced from different B cells. But in order to have just a single type of an antibody, a hybridome is created in this form. Now, what scientists have done is that it's really hard to produce monoclonal in humans, right? I mean, it's unethical to inject a human with a virus and collect antibodies from this person. So what scientists have done is that they have created humanized antibodies by, for example, injecting an antigen into a mouse. And then this mouse would produce antibodies. Now, by taking the gene that produces this immunoglobulin, and it can be with genetic engineering technology, it can be that the concentration would be a human, but the hypervariable regions would be from the mouse. Now, if you inject the mouse immunoglobulin into a patient, that would create an immune reaction because the mouse antibody is foreign. It's an antigen by itself. But the hypervaluable regions are not foreign. They're not considered foreign. So right here you have the production of a humanized immunoglobulin having all sequences and domains of a human immunoglobulin except for the hypervariable regions that recognize the antigen. Now this immunoglobulin, this antibody, can be injected into a human and it can be considered normal, not an antigen. So what are the advantages of monoclonal antibodies? Well, we can use them to measure the amounts of certain proteins and molecules, and you would get the same exact results because the affinity does not change. Okay, so if you do, so now in the clinical laboratories, they use monoclonal antibodies to measure the amount of plasma proteins, steroid hormones, and the same exact results are obtained consistently and reproducibly. Why? Because it's the same antibody binding to the same antigen at the same site and so on. So the other thing is you can determine the nature of infectious agents, right? So by using monoclonal antibodies against a certain type of bacteria, if the bacteria bind to this immunoglobulin, then specifically, of course, then you can say that this bacteria exists in this sample. Monoclonal antibodies are used therapeutically, for example, against tumor cells. So we have the immunoglobulins targeting tumor cells. Now it's actually used to recognize cytokines or inflammatory proteins as a new strategy of treatment. Or, by the way, it can be monoclonal antibodies can be used to remove drugs, to clear drugs from an individual bloodstream.